very useful for us. And so I think you need to evaluate both lecturers and then your TA for your lab section. Please do it. If we don't have enough feedback, we're going to have to do it during class when we're supposed to have the back and forth review session. So it's going to take away time from your being able to ask questions. Okay, so please do that on your own time as soon as possible. Okay, so now we're going to talk about how you screen, generate mutations and screen for them in what we call forward genetics. The real advantage to forward genetic screens, if you're interested in knowing how something works, is you come in with absolutely no bias. You don't think, oh, I think I know how this works, so I'm going to mutate this gene that I think I know has a function here. Instead, you just say, okay, I'm just ready to accept anything that I can find that affects this process. And many times, it's quite surprising what you find and gives us insight into things that we really didn't even know we needed to know. So forward screens are still an extremely important part of how we learn how the biological world works. In a forward genetic screen, what you're doing is you're mutagenizing an organism, hoping to mutate something that's affecting a process that you're interested in. Generally, you try to induce quite a lot of mutations and then have a clever screen to screen for them. Again, you want to basically try to saturate. So not all of your progeny are going to have a mutant phenotype, but some of them will. Now what you do is take the mutants and make them to wild-type females. And if, as you hope, this is a genetically transmitted trait that you're looking at, then half of the progeny should show the same mutant phenotype as that parent. Okay? Simple Mendelian genetics that you are now very familiar with. Thing to realize and this is where I'm saying mutant phenotypes means what is really determined by what you're following. So, for example, if you're looking to see if there are big differences in the body plan, you'll see that beam shape, you'll see that eye color, you'll see that. But say what you've mutated is a gene that affects a behavior. We talked about the circadian clock in the flies. Mutants in ficus, you see a mutant phenotype. That's what we mean by dominant, right? It's not so easy if you generate recessive mutations, and most mutations are recessive. Okay? Dominant mutations are in fact pretty rare. So most of the time what's happening is that you mutate this male, and again, a bunch of its sperm are mutated. And then some of the type fly, if I want to really analyze that mutation. And that's because there's a chance, maybe a good chance if I've done saturation and mutagenesis, that whereas I'm revealing that one mutant, there may be other mutants that are simultaneously being carried in my fly. If I keep mating back to the wild type, but select for the one mutation I'm looking for, in that way I can start to breed away all other mutations so that I have nothing but wild type. Okay, so once you have your mutation, you still have to do what in this class we actually talked about as test process, which tests to see whether something was heterozygous, right, or homozygous dominant. But the same idea is that if you already know you have an F1, if you keep back crossing to the wild type, you can get rid of the other mutant alleles in the background, what we call the genetic background of the fly or whatever organism you're talking about. mated to a female that contains this balancer chromosome called CLB. What I'm going to end up getting is, from this cross between these two, is one type of female I will get is one that has the balancer chromosome and this putative mutagenized X chromosome from the male. Okay? So I've mutated the male, and I'm now going to follow the X chromosome coming from that mutated male. So it's mutated sperm carrying that X chromosome. Of course, it's going to only be found in the females of this generation, right? The male's giving its X, and the female's giving her X. Now, the way I know that this is the fly I'm going to be using for my next cross is the balancer has a dominant marker gene, bar, okay? So I know that the bar-eyed females are the ones I'm interested in. Quick, easy screen. I mean, I just look at them and say, okay, bar eyes, I want all of these, okay? And then chromosome, it has a recessive lethal gene. So if you have that recessive lethal gene next to a Y, it is revealed, and those males die. Okay? So I don't even need to look at these. I didn't want to look at these because they're unimportant to me. They're not interesting to me. What I'm interested in is only males that might be carrying my mutated chromosome. And in fact, the only thing that will survive, it's because the balancer chromosome with half of the Y chromosome, half of the progeny, had the balancer chromosome. And the other half had my mutant chromosome that happens to have a lethal mutation. As a result, all I have to do is look at this F2 cross, and if I have no males at all, I know that back here is a lethal mutation that I can now follow up. Whereas if I do get progeny, now I should just point out that whereas that was the first balancer chromosome generated, there are now balancer chromosomes for the other 3 autosomes in flies. So people are able to screen very rapidly for mutations on any of the Drosophila chromosomes. Another type of organism that's great for finding mutations is a haploid organism. Because now you don't have the problem of dominant versus recessive mutations. Because they only have one copy of every gene. They're haploid. Temperature sensitive mutation. Okay? So one kind of conditional mutant is, for example, this mutation is no problem as long as you give the organism all of the necessary nutrients and add in all the amino acids that they may need. Okay? But if you take away some of the amino acids, most wild-type organisms will grow, but maybe a mutant won't. So you can first put your mutant colonies onto media that has full growth media available, and then you can replica plate them into media that is more restricted, and in that way identify genes that are important to the production or the ability to survive in the absence of that particular enzyme or amino acid or something of that sort. So the other thing is temperature sensitive, and that in fact, just as an example, we refer to when you're doing one of these conditional type screens, you have what is a permissive condition. In the example I just gave you, you give them all of the nutrients they need. And then you have the restricted condition. Say you get rid of methionine, okay, and suddenly they can't survive. Okay? That's the restricted condition. So this brings us back to something that, again, Professor Loiza talked to you about, and that is cell cycle. Okay? Various aspects of the cell cycle. and that in fact much of what we understand about the cycle, that is DNA synthesis and replication and so forth, was in fact determined in yeast, doing mutagenesis of the yeast, but then screening for mutants in different temperatures. Okay. This is just the overall strategy, is that as you treat the yeast with the haploid yeast with a mutagen at what is your permissive temperature, 23 degrees centigrade. And then you plate these out on a plate at 23 degrees centigrade, so that the reason you're plating them, you're putting fewer colonies per plate, so that you can, or fewer cells per plate, so that for every individual yeast cell, that is, of course, they were all subjected to mutagenesis, but not all of them are going to mutate, okay? But you want to look at each individual yeast cell. So the yeast cell falls down on the plate, and it begins to grow and form a colony. If you induced a lethal mutation that is not even able to survive at 23 degrees, you're not going to see a colony at all, okay? So the colonies you see are yeast that can still grow at this permissive temperature. Now what you do is you replica plate them, meaning you take a little bit of each of these colonies and you put it onto another plate, where now you subject the plate to 38 or 36 degrees centigrade. And you may, in fact, identify some colonies that cannot grow. In this particular case, you've transferred not just a single yeast. What you've transferred is what was initially one yeast cell that has turned here, that has generated a bunch of daughter cells, right? So you have a lot of these cells. You've transferred a good number of them over to this new plate, and you're finding that they will not grow, okay? They just, whatever you put there stays there, nothing else happens. But then you can look at the phenotype of those cells and ask, what's wrong with them? Why are they not dividing? Why are they not making progeny? And in fact, in looking at those phenotypes, what they looked like when they were arrested differed from mutation to mutation to mutation, and ultimately this led to an understanding of these sets of genes that were involved in various checkpoints in the cell cycle. So when you talked about checkpoints earlier in the semester, this is in fact how those genes were identified. When I talk about generating a mutation that's temperature sensitive, so the organism is perfectly good at the permissive temperature, but it's not good at the restrictive temperature, would you imagine that I've generated a null mutation? What do I mean by a null mutation? It makes no product. That would not work, right? If you generate a null mutation, there's no product to be made. So when you're at the permissive temperature, you've still got a problem. The idea here is that you're making a mutant protein that, in fact, is not quite as good as the wild type protein. And as a result, when you put it at high temperatures, it's unstable and unable to do its job. So in general, what you're looking at here are changes in the protein rather than a loss of the protein altogether. Okay, another major area of our strategy for understanding various processes, in fact, is to introduce genes from one organism into another. This we call transgenics or transgenesis, and we can mean that we're taking just from one individual mouse and putting it into another mouse. Or we could be taking from a human and putting it into a yeast. are a bacterial gene into designing those changes, whereas nature was doing it for millennia before us. But the introduced gene then is called a transgene. And what we want to do is get it into the genome. Now, in this particular case, we're talking about integrating into the genome, but we want it in a form that it can actually be expressed. So obviously, if we're going to make a transgene, we want to put it into a vector. We'll now know what vector means. So a vector that will either allow the gene to be expressed in prokaryotes, such as this one, or in eukaryotes, such as this one. And I think I already asked you in the last class, why are these different?
One's eukaryotic, one's bacterial.
One's eukaryotic and one's bacterial, but why? Exactly. The regulatory sequences are quite different. So what you need, the regulatory sequence is needed for expression of a bacterial, of a gene of any kind in bacterial cells are very different from the regulatory sequences that you need here. And I think we already talked about the Shine-Dalgarno and the fact that you need to have the Shine-Dalgarno in your vector here because it lies upstream of the AUG. It's defining the next AUG as being the one the ribosome should start translating from. And again, didn't we discuss this as well? Why don't we need the Kozak sequence in this? Because it surrounds the AUG. So that will be in the piece that if we're introducing a gene, the Kozak sequence will be part of the gene because it surrounds the translation start. In fact, the kind of regulatory sequence we put into our transgene is going to dictate how that transgene gets expressed. We can put regulatory sequences that will make the gene express constitutively, just always be on. We can choose a promoter and enhancer sequence that's going to make it only expressed in one particular type of tissue. We can set it up so that we get inducible expression, where it's not on most of the time, but we can add something and turn it on, or we can have it on and add something and turn it off. So all of these things we've been learning about promoters and enhancers, we take advantage of that information to help us create the kind of transgene we need in order to ask the question we want to ask. An example just given here is going into bacterial cells. We can put genes under the control of the lac operons so that only when we add the appropriate inducer, lactose, are we going to get the gene expressed. And then we can get it up to very high levels. There are some problems in moving genes from one organism, from one species or whole animal group to another. And one of those is codon usage. You should be getting excited. You know all this language, you know what we mean by codons, you know, you're really there. So in fact, what you find here, this is very interesting. In E. coli, glycine, of course, on our little chart that we look at, glycine can be encoded. This is a universal code that any one of those triplets will give you glycine. However, in E. coli, there is no tRNA for that. There's no tRNA with an anticodon for that. And so if I use a human gene where about 25% of all, or 1/4 of all glycine codons are in fact that codon, I'm going to have trouble getting that gene. It will get expressed, but I'll have a hard time getting it translated in the bacteria because the bacteria will not be able to translate that particular codon. So I can mutate. I can go in and investigate, and this is what we refer to as increasing the efficiency of a given gene's expression. So, for example, you guys are working with green fluorescent protein. It was maximized for expression in eukaryotes by doing some changes in codon usage in that gene, so that you get enhanced expression in the human. So you see, for example, whereas in bacteria, this is used a lot, this codon. It's used very little in humans. So in general, there is not as much tRNA available for that particular codon. So you want to modify your gene so that it will work in whatever recipient you're going to use it. There are other complications, and that is particularly in animal cells, a lot of post-translational processing takes place. Here I'm not talking about RNA splicing, and that's post-transcriptional. But post-translational means you do things to the protein once it's made, such as you need to add sugars, you get glycosylations, you get phosphorylations, and these are important for the function of that protein. If that's the case, expressing it in an E. coli may generate a protein, but a non-functional one, okay? Because it can't be modified in the ways it normally would be in the organisms from which it came. But be that as it may, bacteria are so great because, like the fly, they're better than the fly. They, grow up tremendous meters worth of these organisms, and if they will express what you want them to express, they can make copious amounts of protein. You can just harvest the media from these cells, particularly if they're making something that's secreted from the cell. You can just spin the cells down and have liters worth of this protein. And in fact, that has been used. Many of the things we use in medicine and other industrial applications involve genes that have been put into bacteria and expressed in an expression vector so that we can make a whole lot of it, like human growth hormone. I always also refer to the pregnancy test, okay? In other words, there are a lot of things that we use now just over the counter in the drugstore are actually the result of recombinant DNA, where you put a gene into a vector and put it into bacteria, and then you can harvest copious amounts of it. You're saying here we even have them in our detergents now. Enzymes in our detergents. Now, when we put transgenes into bacteria, as you know, we can put them in a plasmid that is autonomously replicating. And as a result, it can just replicate to high copy number, and you can get expression without anything integrating into the bacterial chromosome. But in eukaryotes, in general, what we need is to get the gene integrated into the host genome. And this is important for the gene to stay stable. These plasmids will be perfectly learned from Professor Llewellyn. The frequency with which introduced DNA is either just integrated by illegitimate recombination or by homologous recombination. The difference has to do with what the actual host species. In most plant and animal species, what happens is illegitimate recombination, meaning you're introducing a gene, it just goes wherever it feels like and integrates into the chromosome. Okay? Whereas in yeast and in bacteria, and particularly when we have those long bacteria, those backs, we can get homologous recombination into a back. And homologous recombination into yeast is very easy. So let's just talk about a moment about what kind of process this involves. If you use a circular piece of DNA and you put it into yeast, in fact, yeast can integrate it if you have one crossover, that is one break in your plasmid and one break in the yeast, even though you are this is happening because the plasmid has homology with the target gene. So it's annealing to its homolog, okay? But if you have only one break, this whole plasmid is just going to integrate into the yeast genome sitting next to the target gene, not replacing it. The only way to get it replaced is to have two breaks in both partners, in which case you will, in fact, replace your target gene with this one you're introducing. Now, this is a case where, for example, we have this gene. We want to know what it does. We've cloned it, but we have no idea what it really does in yeast. So what we've done is in the plasmid, we've introduced a selectable marker gene right in the middle of this gene, which means when the gene gets transcribed and it starts to get translated, you get premature stops because it has this weird new sequence in the middle of it, okay? So this is a very common way to, quote, knock out a gene. Introduce some foreign DNA right into the middle of a gene, and you've messed that gene up. It's a knockout, okay? Now, it doesn't always lead to something that can't be used. Yeah?
I just have a general question. How do you get the plasmid into a nucleus in the first place?
How do you get it into the yeast? The
the plasmid has to get inside of the nucleus, right? To be.
Yeah, it does it. It does it.
By itself?
It does it by itself.
Really?
Yes, yes. So we can electroporate. Okay, so we give an electric shock is one way that we get into yeast, and it's the way that we get in even to mouse cells. We just give a shock, it goes in, and some of it gets all the way into the nucleus. Okay, so at any rate, so here's an instance where what we want to do is to replace the wild type gene with this mutant gene, which is basically a null. And now we can say, did that influence the activity or the ability of this yeast to live, or its ability to mate with other yeast, or its ability to do whatever, okay? So the problem is that when you use a circular piece of DNA in yeast, it could either do this or that. On the other hand, if you use a linear, that is just a straight line of DNA, not a circle, a linear piece of DNA, then again, it can recombine with one break in each of the two participating species. But if this happens, you end up with your yeast chromosome crossing over, picking up over here, and this thing is you basically have a bunch of chromosome lost. Okay, because you only have a centromere on one part of the chromosome. So you're going to end up getting a huge deletion, and huge deletions like that have too many genes, and the yeast is just going to die. On the other hand, if you get a double crossover with a linear piece of DNA, you're going to do what you want to do, is get a target gene. So in fact, if you transfect, so we call it transfection with a transgene, if we transfect a yeast with linear DNA, and then we select for the selectable marker, the reporter we put in there, anything that survives is a homologous recombinant. So yeast is great for homologous recombination. And again, it has been used over and over again to identify functions of genes. But yeast don't have a body, okay? They don't have feet and hands and hearts and all these things. So when we need to know about other things besides the things that yeast can do. Yeast told us a lot about cell cycle, for example, as we just discussed. Well, we want to do studies of genes that are important to higher, we don't like to call them higher organisms, but more complex organisms, okay? So, unfortunately, homologous recombination is not at all common. It's not that it doesn't happen, it's just way less frequent than just illegitimate recombination in animal cells. In addition, in animals, we're not single cells, so we have to figure out where to put the gene such that it will ultimately be expressed wherever we want it to be expressed. And the only kinds of cells that are going to give rise to a whole organism, multicellular organism, are the gametes, like eggs, or in embryos, that is, in cells that can give rise to an entire animal. So we're going to have to, in transgenics in an animal, we inject our gene into gametes or embryos, which we'll get to in a minute. Generally, we can't get it injected depending on the organism. But certainly in mice, which is where we do a lot of transgenics, we can't inject right into the egg or into the sperm very effectively. It usually doesn't work. So instead, what we end up doing is putting it into cells that are going to give rise to the organism, and we're going to get a chimera, which I think I'll talk about in a minute so that you understand that better. Okay, so one way of making transgenic mice, for example, is to take a fertilized egg. Okay, so you have a fertilized egg, and it hasn't even started to divide yet. Okay, and you can inject literally with a needle. Okay, you can inject your DNA into that fertilized egg, and now your gene is just going to randomly integrate wherever it decides to integrate. And then you're going to hope that you didn't kill the fertilized egg and that it then divides and gives rise to a whole organism. The thing is that each fertilized egg is going to have that gene in a different place. Okay? So each transgenic animal is different in that the gene that you've given it is the same, but where it's integrated is different. And why would that matter? Think of chromatin. Is chromatin the same throughout the genome?
No.
No, we have heterochromatin, which is not very good for gene expression, and euchromatin, which is. But you can sit down next to a gene that's being very highly expressed, and it has an enhancer that's going to make your transgene suddenly expressed at high levels. Or you could sit down to another gene that has a very strong silencer that will turn off your gene. So depending on where the gene goes, in each individual egg, you're going to have a mouse that is expressing your gene, another one that's not, another one that's expressing at a low level, another one that's expressing at a very high level. Okay, so you're going to be seeing a lot of different possibilities. That means that, and the other thing that happens when you use transgenes and illegitimate recombination in animals is they tend to go in not as a single copy. It's very hard to get a transgene in, for example, mice to go in as a single copy. It usually concatemerizes. It makes multiple copies of itself. Remember, we're not taking a single molecule of DNA. We're taking when I say I'm going to take, or you take a plasmid, all right, or I take a linear piece of DNA, you've got, hundreds of thousands, millions of molecules of the exact same thing, right? They're just a bunch of replicates. You inject them, and they tend to ligate to one another end on end to form what we call a concatemer. They don't all end up going in the same direction in that concatemer, okay? And remember siRNA? That is when you make an RNA that has a palindrome, it's going to fold back on itself. You're creating a palindrome when you have things going in opposite orient, the same thing going in opposite directions. Okay, so getting those palindromes can sometimes lead to siRNA effects on your gene. But also just having multiple copies can mean that you're not going to express your gene at its, quote, normal level. You're going to express it extremely high level. And that may change the what it does. Okay? We already know that. We know that many genes are not haplosufficient. There's the opposite of having, if you express something at really high levels, it can be that's one of the reasons that chromosome things like trisomy are a problem. When you get too many copies of the gene, it's a problem. Too few copies of the gene, it's a problem. So we've learned a lot from transgenics where we've done this. And in fact, that situation of going in anywhere in the chromosome and sometimes therefore being silent led to our identification of that locus control region in the beta-glucan locus. That was a gene that once you added that LCR, in fact, you could look at 15 different mice that had picked up that gene and put it in different places in the chromosome. And in every case, it was being expressed. It also did have the problem of having multiples, but for if you had one copy, you got one level. If you had two copies, you got 2 times that level. If you had 3 copies, you got 3 times that level. And that is, it acted like an independent locus, no matter where it went. So this kind of transgenesis actually led to our discovery of those kinds of regulatory sequences, the LCR. But you can see that there are problems too. So everyone was busy trying to figure out how can we get homologous recombination to happen in mice. Well, this is what was figured out, and that is to use what we call a positive-negative selection strategy. And it's really very simple. You make your transgene, and here we've got an example where we have the CFTR gene, and we're going to do a knockout. So we're going to put a selectable marker, neomycin resistance, in the middle of that gene. So the idea here is going to be that generally mouse cells can't live in the antibiotic neomycin. But if they have a neomycin resistance gene, they can. Okay? So this is going to be our positive selection marker. And in addition, it's going to serve 2 purposes. It's going to be a positive selection marker, and it's going to be a way to, quote, knock out our gene. But the new idea in these constructs was to add this. And again, this is a case where basic biology, understanding what things do, how they work, allows you to piggyback until you can do something you never thought you could do. So this is a case where there's the herpes simplex virus was being studied. It has a thymidine kinase gene, which, like our thymidine kinase gene, is phosphorylating thymidine so that it can be incorporated into DNA. But that HSV-TK, okay, that's what this is, thymidine kinase, the viral form of this enzyme is able to act on a substrate that we and mice and other eukaryotes never use and do. That is, it can use a substrate called ganaciclovir. When it does, what it does is it phosphorylates this as a substrate, and that acts as a mimic of thymidine. And it gets incorporated into the DNA. Wherever we see an A, that gets added as a T. But nothing more can happen. and so it stops all DNA replication. So this is how you can kill the cell, okay? It can't replicate any further if you've got this enzyme running around and you've got this substrate, okay? So that's our negative selection marker. So here we take this linear piece of DNA and we inject it into, in fact, this is a case again where we actually do things like electroporate into cells. What we use in this case are embryonic stem cells. They're derived from embryos. It's basically taking a mouse embryo and breaking it apart into single cells. And all those cells are pluripotent. They're able to generate all kinds of tissues. We now have such cells that we can keep frozen in the freezer, and anybody can pull them out. You can buy them from a company, these embryonic stem cells for mice. And we're going to use those to knock out this gene, because in each one of those individual cells can make a mouse. Okay? So we're taking those cells and we are introducing this new positive-negative selection vector into them. And what you see is that three different things can happen. One is our transgene does not get into the DNA at all. So here's the transgene, here's the recipient ES cell chromosome, which is in fact a mouse chromosome. Nothing happens. And in cells like that, if you plate the cells after transfection on plates that have both neomycin and ganciclovir, they die because they are neomycin resistant. They don't have any resistance to that antibiotics. They all die. Alternatively, our transgene can undergo illegitimate recombination, so that in this case they're showing you a situation where there is a double crossover. And you are replacing DNA, but you're not replacing DNA that has anything to do with that gene. You're just going in somewhere into the chromosome. Okay? In that case, you have integrated both the neomycin gene and the ganciclovir gene. Sorry, the HSC-TK gene, the TK gene. So now these cells are neomycin resistant, but since they have that TK gene, they use ganciclovir, and that stops DNA replication, and so they die. So we call them ganciclovir sensitive. They die. So what lives? What lives is a case where we've got homologous recombination between the transgene and this chromosome. So you get recombination between the homology here and the homology there. And so if you're looking at the mouse chromosome, you're going up here, picking up that mutation and coming right back down, and you create a new chromosome that has the mutant gene. So by this kind of selection, we select for homologous recombination. And we always have to check it. We do all kinds of checks after doing that, because this selection is really great but not perfect. You always have to make sure that in fact you haven't, that you are sitting right in this site. And I won't get into, you can use PCR to convince yourself of that. But at any rate, we can get homologous recombination in animal cells. So now you take those embryonic stem cells that you put on neomycin and ganciclovir. And one thing to remember is that these are diploid organisms. So you have now modified one copy of your target gene, but the other copy is just fine. Okay, so it's a heterozygous mutant. And now this is where those ES cells, you cannot just take an embryonic stem cell and put it into the uterus of a mouse and get anything to happen, okay? Because of course they came from an intact embryo, okay? They are not able to form the whole thing by themselves. So instead what you do is you take a female, and what you're going to notice here is that we're taking a particular genetic we're using marker genes that will be important to our figuring out when we have generated a mouse from that ES cell that has that mutation we just generated. Okay. So what we're going to do is take a female that's wild type, of course, for our gene, and it actually is homozygous little b, which makes it a black mouse. The embryonic stem cells we're using, however, as we said, it's heterozygous for this mutation, but it's also, though embryonic stem cells were taken from a homozygous big B, which is a brown mouse. So from this female, it's pregnant female, and it has got some embryos growing in it, or it's got fertilized egg growing in it. And once an egg is fertilized, it begins to divide and divide and divide until it forms a hollow shell of cells, which we call the blastocyst. We take up the blastocyst, and as you know, mice don't just have one baby at a time. They usually have 5 or 6. So we can take multiple blastocysts out of a single female. And in those blastocysts, we're going to inject those embryonic stem cells that we have confirmed have that mutation. They're going to get incorporated into this blastocyst, and there are going to be some of the tissue, they're going to grow into a little, a big enough thing to be born, and then grow into a whole mouse, where some of their cells come from the ES progenitor, and some of the cells come from that original mother here. Okay. So we take the blastocyst, we inject with our ES cells, we re-implant, we actually don't go back to that mother, but we implant into another female who's been treated with hormones so that she's ready to have embryos grow in her. So she gets injected with a bunch of different blastocysts, all of which have been injected with the RES cells. And if you're lucky, she's going to give rise to little pups. And I hope you can see that these pups are not brown like these are. They are mottled. They're black and brown. In this case, our experiment has worked. In reality, you get a bunch of black pups from blastocysts that never incorporated the ES cells, and then you get a few mottled babies that look like this, and you get very excited when you see the mottled ones. Okay? So you say, okay, what this is telling me is that this is a genetic chimera, meaning that literally there are cells in this animal that I like to say some of them have Maria's genome, that is the genome that came from the ES cells, and other cells have a completely different genome, Sandra's genome, that come from that mother that was the blastocyst of that mother. Okay. So what that means is that the germ cells that are going to develop in those animals, some of them will have the ES genes. So let's talk about a sperm. If we have the precursor to the sperm that actually has been developed from the ES cell, then they have our heterozygous mutation, right? They've got the whole genome of the ES cell, including the big B, right? And including this CTFR gene, both a wild type copy and a mutant copy. Okay. So when we make sperm from that cell, some of the sperm are going to have a big B and the mutant gene, right? And some cells will have a big B and the wild type gene. So when we take these mice and we make them back with this mass, little b, if the ES cells have gotten into and formed the germline in this animal, we're going to get some pups that look like this. Okay? That is, they're brown. And the reason they're brown is because they have the big B coming from the ES sperm, and they've got, and of course they've got the little b coming from this mother. So they're heterozygous B, but they have a chance of having our mutant gene here. Yeah.
Why do we use a different method?
Say that again?
Why do we implant the blastocytes into a different mom? Why do we not put them back into the original
Mother, because oftentimes that mother has to be sacrificed to get all of those blastocysts out. Okay, so we put it into an intact female later. Okay, so I don't know that what you're going to look for then when you do this mating is brown progeny, which is going to tell you that for sure you have picked up sperm from the ES cells that have the ES genes in them, okay? What you see in this diagram is two such mice that are brown and that are heterozygous for this gene. Is that always going to be the case for every brown mouse you see? No. You're going to get the little b and the big CTFR always from this parent, right? But what you get from this parent could be the big B and the mutant, or the big B and the wild type. So you can have brown mice that don't have your mutation. So you're excited you got these. You're ecstatic you got these. Because many times what happens is you do this mating and you never get any brown mice. And the reason for that is that it never went into the germline. The ES cells never ended up generating the tissue that was going to give rise to the germline. So that's not uncommon for that to happen. But if you get these browns, you're going to now have to do further matings to figure out which ones are carrying your mutation. And we do that directly, many times with just, again, DNA PCR. But once you've figured out that this is carrying the mutation, perhaps by PCR, and you can imagine in your heads how we could figure that out. I'm just going to look for a gene that has neat CTFR sequences that when you use primers around the insert, you see neo, okay? So at any rate, you can identify which of these progeny do carry the mutation, and if you mate them with one another, you're going to end up with homozygous knockout mice. Now, sometimes we want to manipulate the gene once we put it in. Maybe we want it, say in fact, maybe we don't even interrupt the gene. We just want to be in a situation where we can get rid of it at a certain time in development, or get rid of it only at when we add an inducer of some kind, okay? And we can do that because, again, of looking at model organisms, understanding microorganisms. In this case, it's not even a microorganism, a bacteriophage. We take advantage of the biology we learn by studying those various types of organisms, like the bacteriophage. The bacteriophage, we know, goes in and integrates itself into the bacterial genome, and it does it in a very specific way. We can harness that to our purposes. So an example of that is the P1 bacteriophage, which goes very specifically into DNA sequences that we call the LOX-P sequence. And in fact, what happens with lots is that the bacteriophage is looking for this sequence, and the bacteriophage is making this enzyme called pre-recombinase. But we can use that system. What the pre-recombinase does is it causes the LOXP that's in the bacterial genome to interact with the LOXP that's in the bacteriophage so that the bacteriophage can integrate into, get recombination between the two. But we can put two LOXP sites in our genome, and if they're in the same direction as shown by these arrows, which means the sequence is in tandem, okay? Then when we add pre-recombinase, we can cause recombination and get something to loop out of the DNA. So we can cause deletions anywhere we want just by introducing these LOXP sites into areas where we want to do something. So in that example we just gave, we could have the neo gene there, or maybe we wouldn't have the neo, well, we would have the neo gene somewhere in order to do selection. But we put LOXP sites around that CTFR gene. And so when we got homologous recombination to work, we now have a new gene that's flanked by LOXP sites. And we could induce deletion of that gene at any time we wanted by expressing the pre-recombinase. Just as an aside, if you put the LOXP sites in opposite orientations to one another, you will not get deletion, but rather you will get inversion. And in some cases, that's what you want to do. But we don't make pre-recombinase, and neither do mice. So you have to provide the gene for pre-recombinase. You can do that as just a regular illegitimate recombination event, making a transgenic animal that has the pre-recombinase. But what's really cool is that you can put that gene under the control of a tissue-specific promoter enhancer, for example. So you can say, I only want pre to turn on when B lymphocytes get to the stage of Ig secreting cells. And I want to see what happens if I delete immunoglobulin genes at that stage. Or, you know, I want to go into neuronal cells, and I only want to delete this gene in a particular type of neuron. There are lots of neurons in the brain. We don't know what they're all doing. Let's get rid of this gene and only a subset of those neurons. Well, I put pre under the control of a promoter enhancer that works only in those neurons. Okay? So there's a lot of manipulation that we can do to follow with this system. So how do we use the pre system? I just told you. We can use it so we knock it. Sometimes if we knock out the gene, like we just did, and do the homologous recombination, we'll never get a mouse because it's an embryonic lethal to knock out that gene. That is, the mouse needs it to develop. But it turns out that once you get past a certain developmental stage, or it's doing something else in other cell types, so you can wait and delete it later in development to see its function. One thing I'm sorry that we're not able to cover in this course is plant transgenesis, because this is GMO, genetically modified organisms. And I urge you to read the chapter about that so that you're more knowledgeable about it. You can read it, you can understand it. You've got the language, you've got the techniques. But one of the things that people working with modifying corn, for example, do is that they want to get rid of those antibiotic markers that they use, those selectable markers they use to get things into the genome. And they use the pre-recombinase system to get rid of it. And all they leave is like a little 6-base pair. I can't remember how many base pairs, but the LOXP sequence is quite small. That's all that's left in place of where that marker was. Also, people like myself who work with regulatory sequences within the genome, we found that when we started to try to knock out those, like knock out enhancers, and of course we were introducing a marker gene in there, that we couldn't really look at the clean effects of the loss of our regulatory sequences because we were putting in an expression that, we were putting in neomycin resistance, for example, under the control of a eukaryotic promoter and enhancer. So we needed to get rid of it so that it wouldn't replace the function of the sequences we were trying to get rid of. So this Cree recombinase allowed us to cleanly delete regulatory sequences and study their function. Okay, I'm 3 minutes early. That's the end of today's lecture. I have posted on Blackboard sort of a listing of the material that we've covered in these 4 lectures, what you should be reading in order to study for the final. There are methods in there that I've already told you I want you to read, but look very carefully at what I tell you there, as I also outline for you the subjects that we haven't covered and that will not be covered on the exam. Okay? So I'll see you a week from next Tuesday. Stand for questions.
Hello.